Good day, this is Professor Will, CBMD PhD. Today is Thursday, November 25th, 2021. It is 7:57 and 7 seconds, a.m. Eastern Standard Time. Today's research, intestinal stem cells. So let's hop to it here. There is a new link between diet, intestinal stem cells, and disease discovered. Date November 19th, 2021. Source, German Research Center for Environmental Health. The intestine is essential for maintaining our balance. It is a master at reacting quickly to changes in nutrition and nutrient balance. It manages to do this with the help of intestinal cells that, among other things, are specialized in the absorption of food components or the secretion of hormones. In adult humans, the intestinal cells regenerate every 5 to 7 days. The ability to constantly renew and develop all types of intestinal cells from intestinal stem cells is crucial for the natural adaptability of the digestive system. However, a long-term diet high in sugar and fat disrupts adaptation and can contribute to the development of obesity, type 2 diabetes, and gastrointestinal cancer. The molecular mechanisms behind the maladaptation are part of the research field. The scientists assume that intestinal stem cells play a special role in maladaptation, and using a mouse model, the researchers investigated the effects of a high sugar and high fat diet and compared it with a control group. The first thing researchers noticed was that the small intestine increases greatly in size on a high calorie diet. Researchers then profiled 27,000 intestinal cells from control diet and high fat, high sugar diet fed mice. And then, using new machine learning techniques, researchers thus found that intestinal stem cells divide and differentiate significantly faster in the mice on an unhealthy diet. The researchers hypothesized that this is due to an upregulation of the relevant signaling pathways, which is associated with an acceleration of tumor growth in many cancers. And this can be an important link. Diet influences metabolic signaling, which leads to excessive growth of intestinal stem cells, and ultimately to an increased risk of gastrointestinal cancer. With the help of this high-resolution technique, the researchers have also been able to study rare cell types in the intestine, for example, hormone-secreting cells. Among other things, they were able to show that an unhealthy diet leads to a reduction in serotonin-producing cells in the intestine, and this can result in intestinal inertia, typical of diabetes mellitus, or increased appetite. Furthermore, the study showed that the absorbing cells adapt to a high-fat diet, and their functionality increases, thus promoting weight gain. What researchers have found out is of crucial importance for developing alternative non-invasive therapies. And today, there is no pharmacological approach to prevent, stop, or reverse obesity and diabetes. Only bariatric surgery causes permanent weight loss and can even lead to remission of diabetes. Novel non-invasive therapies can happen, for example, at the hormonal level, through targeted regulation of serotonin levels. The research group will examine this and other approaches in subsequent studies. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.